those that are being killed by your gut, will actually cause an endotoxic response. And these kind of bacteria are deemed enteroinvasive, so they have the ability to invade. So very often, different strains of bacteria will carry different plasmids, and the symptoms of the infection that you have with the particular pathogenic E. coli is often dependent on what plasmid they're actually carrying. So remember, the E. coli that are in our gut are not pathogenic per se. They are natural flora of our gut. We don't carry usually pathogenic strains of E. coli to a large extent, but some people do. But contaminated water can ultimately lead to these pathogenic E. coli. So very often one of the common places you get E. coli, these kind of E. coli infections are due to eating contaminated food or drinking contaminated water. Remember, however, if we take those same E. coli that are in our gut and put them somewhere else, like if in the process of living your normal life, you do not carry out proper hygiene, you can easily result in a bladder infection, or which can then lead to a kidney infection, which is really an infection caused by your own E. coli cells from hygiene that is not properly carried out. So if they go somewhere else, they do cause infections. So hence, oftentimes when you're having surgeries, it's not just the external E. coli or the external bacteria of the environment that could cause problems. It's actually the bacteria that's actually in your body that ends up going in an incorrect place, like a person who actually is having abdominal surgery and they end up with a perforated gut. And all the material in their gut spills out. That includes all the bacteria, and that leads to a systemic infection very quickly. Okay, so some examples of pathogenicity and virulence due to the presence of a plasmid or a virus is listed here. And you can start seeing that many of the classic infections / diseases we talk about as being major bacterial infections are actually caused by the bacterium carrying a plasmid or a lysogenic virus. Okay, so remember, viruses and bacteria can be lytic, in which case they kill the bacteria, or viruses can be lysogenic, which means what? If bacteria is carrying a lysogenic virus, what does that mean? Where has the phage DNA gone? Yes, so it's integrated into the host DNA. It's quiescence in terms of causing a viral infection, but it's DNA that's being carried in the genome. So, for example, the anthrax toxins that cause anthrax are actually carried on a plasmid, and they're cytotoxic. A plasmid also carries a tetanus toxin, which is actually a neurotoxin, and we'll talk about a little bit how the tetanus toxin works in a minute. It's actually a virus, lysogenic virus, that encodes the botulinum toxin. It causes botulism, and of course is what people inject into their skin to reduce wrinkles. That's Botox. They're actually injecting a bacteria that has a lysogenic, is a toxin that's been released by a bacteria that has a lysogenic virus. Diphtheria is also caused by a lysogenic virus. It causes diphtheria, and it's found in Corynebacterium diphtheriae. And it's actually a plasmid that encoded a toxin that is responsible for the plague. And one of the other things that you have to understand is that on these plasmids or lysogenic viruses, these factors may actually be within a transposon. So the transposon is part of the plasmid or the virus, which we also know means that ultimately that could then jump also to the chromosome itself. So the virus, for example, that encodes cholera toxin is a classic AB exotoxin. So regardless of whether it's encoded on a plasmid or a lysogenic phage, whether it's made by gram-positives or gram-negatives, whether they're aerobes or anaerobes, most of the ones that I've listed here are examples of the classic AB exotoxins that bacteria make. So this is a major class of exotoxins. And as its name would suggest, there's an A subunit and a B subunit. So there's two subunits to this protein. So it's a heteromeric protein because it's composed of two different subunits. And within the AB exotoxins, there's subclasses called the enterotoxins that affect intestines, which of course would be examples that are encoded by the antiplasm. So basically, there's two polypeptides that make up this toxin. The B polypeptide is actually part of the toxin that binds to the host receptor. So again, we have a host cell with the nucleus and its mitochondria, and we have receptors on the surface. And then we have the AB exotoxin that's been secreted by our bacterial cell. And it's the B component that actually recognizes the correct receptor to the host. So it's the B receptor that's targeting it to a particular host. Once the B is interacted with the receptor, then the A polypeptide then enters the cell, and there's a variety of mechanisms, but for our purposes we'll say it enters the cell. It is the one that has the enzymatic activity. It's the A subunit that causes the actual damage that particular exotoxins cause. So for example, the botulinum toxin or Botox toxin, the B component binds to the gangliosides on presynaptic neurons, and then the A component enters, and it's actually an endopeptidase. So endopeptidase means it's a peptidase, so it breaks down proteins, and it's an endopeptidase, so it cleaves within the sequence of generally a protein, so it's an endopeptidase. And it will cleave proteins, and it specifically targets the protein required for the neurotransmitter acetylcholine. So basically it prevents the acetylcholine signaling from taking place, and this results in a flaccid paralysis, if you will. So Botox then is in fact probably one of the most serious toxins known. It can cause death very quickly, and now it's being used therapeutically. I don't consider removal of wrinkles therapeutic. It's cosmetic. But it has been used therapeutically to help people with things like migraine headaches, where the belief is some migraine headaches are caused by some spasming. So by using the Botox, you can actually cause the paralysis of that signal, and as a result, you reduce the muscle spasms. It's also been attempted to help with cerebral palsy symptoms, and of course cosmetically with wrinkles. Hence why if people have not great Botox or too much Botox, they go to somebody who's not really trained to give it, their face looks like they can't even smile, because literally all their muscles in their face have been frozen. So very often some people actually, it goes to an extreme where they can't move their eyelids, they can't smile, because fortunately it's short term, but it will last for a period of time, and then the toxin will wear off. So if that's not a minor point, those people who choose to have Botox need to go to people who are certified to properly deliver it, because there are many people doing Botox that actually are not certified to do it, and you need to know where they're getting the Botox from, because they could be getting impure forms of the Botox. So how does some of these various toxins work? So this is cholera toxin, so this is an enterotoxin. So enterotoxin means it functions where? Enterotoxin functions in our, in our gut, in our intestines. Okay. So normally in our intestines, so here's our intestinal cells, here's our blood, this is the lumen, there's a receptor that's on intestinal cells. Normally sodium enters from the lumen through the intestinal cells into the blood. But if you get a colonization and toxin production by Vibrio cholera, so here's some Vibrio cholera, the classic comma shape, it starts to secrete its AB exotoxin, that's the green or red protein. The B, remember, is the subunit that binds to the receptor, so the cholera AB toxin, specifically the B form, binds to the receptor. What this does is the A enters, and then the A targets adenyl cyclase. So for the purposes of us understanding, because this is a signal transduction course, basically when that targeting takes place, there's an alteration. Adenyl cyclase controls the conversion of ATP to cyclic AMP, and this change in signaling basically blocks the entrance of sodium and results in a net movement of chloride and bicarbonate out of the cell. Okay, so suddenly now, instead of sodium coming in, chloride's going out. There's a net massive movement of these things out of the cell. Sodium is also accumulating outside the cell. So what do we know about osmosis in liquids? Things want to move to equalize. So in essence, what happens is, because of this, water is pulled out of the bloodstream into the lumen to try and equalize the salt concentrations. So basically, you start losing huge amounts of water from your blood. Okay. And so this leads to a loss of fluid in the intestines. You end up with severe diarrhea. And for people who die from cholera quickly, you die from, again, diarrhea and loss of fluid. So that's one classic way an AV exotoxin works. This is actually botulism here. This is tetanus here. Basically, there are two sides of a coin. They're both neurotoxins. So normally, here's an excitatory signal that's coming through the central nervous system. Acetylcholine is critical to that. This is a muscle, and the acetylcholine causes the muscle contraction. In the case of botulism, what ends up happening is the toxin actually blocks the receptors, and as a result, contraction is inhibited. So you end up with flaccid paralysis. None of your muscles can contract. That's why the wrinkles go away. In the case of tetanus, it's the opposite scenario. It affects the inhibitory neurons. So normally we have inhibition by glycine. So glycine is released, and it basically stops acetylcholine from being released and allows the relaxation of the muscle. In the case of the toxin, the tetanus toxin, basically it binds to these neurons. It prevents the release of glycine. So now your muscles are stuck in a contraction. Hence why often tetanus is referred to as lockjaw, because you actually can't open your mouth. Your muscles are forced into maintaining a tight constriction. And as a result, you again end up not being able to survive, because that ends up happening to most of your muscles related to any muscles that are relying on nervation, and that's all of our muscles. So in essence, all your muscles become contracted. So this is just an example of a variety of organisms, a variety of diseases, and I talked to you about a few of them, and a variety of types of toxins and the kinds of actions they cause. So another interest, so you're responsible for the ones we've talked about, but you can see some of the other classic things like whooping cough, caused by Rotella pertussis. So whooping cough, as its name would suggest, is a cough that sounds terrible. So if a small child has it, they make a whoop when they cough because they have trouble moving oxygen. And it's really because of the AV exotoxin, which basically blocks G protein signaling and basically causes the cilia to stop moving. And it causes the inability to move oxygen. So you can see there are many types of toxins. Many of them are AV-type toxins, whether they be specifically causing problems in the gut or in a general toxin. So bacteria rely on these toxins to cause many of the symptoms that we classically think of as a bacterial infection. So some other types of exotoxins include membrane-disrupting exotoxins. These are not AV. They don't have AV subtypes. In general, there are two types. There are those that cause pores to form, and there's those that are phospholipases. So phospholipases would attack who? An enzyme called phospholipase is going to attack what structure? Phospholipids. Where do we find phospholipids? In membranes. So an enzyme is breaking down phospholipids is going to significantly damage this membrane or damage the phospholipids with creating channels or disrupting membranes. So we'll look at a couple of these actually in laboratory later in term. So a classic example of a membrane-disrupting exotoxins that are produced by Staphylococcus and Streptococcus. They actually produce white blood cell killing toxins called mucosidins, which can kill phagocytic cells. And basically, it's an enzyme that actually forms pores in the plasma membrane. And not only can they kill white blood cells, but they can also destroy red blood cells as well. So basically, these exotoxins cause white blood cells and white blood cells basically to lice. Okay. And there's a couple of hemolysins that are produced specifically by Streptococcus pyogenes. One's called streptolysin O, which is actually inactivated by oxygen. Another one that is oxygen stable called streptolysin S. So in lab, when you actually do your blood agar plate, in it's week 8, you actually take a swab of your throat. So you take a cotton swab of your throat, not your mouth, and you put it onto a blood agar plate to ask, do you have streptococci and staphylococci in your throat that can destroy red blood cells? So the blood agar contains red blood cells. If it can, it'll either cause it to clear, so you'll get a clearing zone around the bacteria, or you'll get a greenish tinge around the bacteria. So what would the greenish tinge around the bacteria mean? What do red blood cells carry? Okay, iron bound to what? Hemoglobin. So the release of that molecule causes the red blood cell color to go green because of the release of hemoglobin. So we will actually test this in lab. There's also examples, as I said, of phospholipases, which specifically attack the eukaryotic host cytoplasmic membrane. Then there's another enzyme called streptokinase. And by the way, these are just a small example of the types of disrupting toxins that are available to the bacterial arsenal. Streptokinase is produced by both Streptococcus pyogenes and Staphylococci species. We will talk a lot about Streptococcus pyogenes and Staphylococcus aureus, two of the very nasty gram-positive bacteria that cause significant infections. And they have a lot of arsenals of enzymes to do this. Streptokinase actually breaks apart fibrin clots. So basically it binds to plasminogen that forms at the site of infection and breaks the clot down. So part of our way in which we fight infections is your body forms blood clots around a cut. Not only does that obviously cause importance so you don't bleed to death, but it's also important because it walls off infections. But if a bacteria can secrete an enzyme to break down blood clots, it now can move into a systemic infection. We'll come back and talk about this in a lot more detail. But streptokinase is important in these highly pathogenic bacteria to allow that movement. We have hijacked streptokinase. Does anybody know for what purpose? And this is again an example of how medicine can make use of natural products. So where might you want to break down blood clots? In the treatment of what? Stroke. So actually that injection that you get within a half an hour to an hour, or I think up to 3 hours of having a stroke. So you throw a blood clot is actually an injection of a variation of streptokinase to break down the blood clots. So here's an example of one such pore-forming cytotoxin. Just to give you an example of what happens. This is staphylococcal alpha toxin. So basically what it does is the toxin can actually come together to form a pore in the plasma membrane of the host cell and basically can result in the efflux of cytoplasmic components and the movement of other molecules into the cell, basically leading to complete disruption of the cell. All right, so before we finish plasmids, I just want to briefly talk about engineered plasmids. Then we will be done with plasmids, and that will be to where we'll be at the point of your midterm. Okay, so as I told you, engineered plasmids are the plasmids we design to carry out biotechnology, molecular biology experiments. They're the vectors that are used in cloning, gene expression studies, etc. They are constructed in the library, in the lab, and they have all the things that have made the plasmids that we've talked about nasty removed. So basically, we maintain the origin of replication in these plasmids, but we remove all these bad genes and the ability to be from a perspective of infection and to be able to carry out conjugation. So they're non-conjugate. So in order to get them to get into a cell, we need to carry out a laboratory-based transformation that adds calcium to alter the charge on the cell wall. So what's the charge on a bacterial cell wall? Negative. So we have a bacterial cell, say gram-negative, because with its outer membrane and its inner membrane, it's periplasmic space. This is E. coli, the classic organism used in molecular biology. It has negative charges, and that's due to the phospholipids, actually, that are in the outer membrane. So we add calcium chloride that's positively charged. It masks that negative charge. What's the charge of DNA? Plasmid will have a, what charge? A negative charge because of the phosphate groups in the DNA. So the presence of the calcium masks the charge and gives a surface on which the DNA can bind. So there's a transient interaction of the DNA based on charge to the calcium. So adding calcium makes the bacterial cell competent to take up the DNA. The DNA binds, and then we add a short heat shock and the DNA transiently enters. You will do a transformation in lab, and you'll also do a natural conjugation in lab. So you compare conjugation versus experimental transformation. So in order to get any bacterius E. coli to take up an engineered plasmid, we have to use either calcium chloride or an electric current to cause the DNA to enter the cell. So to be useful as a cloning vector, the vector, i.e. the plasmid DNA, must have cloning sites, which is called a multiple cloning site. This is not a minor point. That means that in order to be a cloning site, you can only linearize the DNA. So I want to clone a piece of DNA. So I have a plasmid. I want to clone a piece of DNA. So I want to insert a piece of DNA right here. I can use a restriction enzyme to do that, but that restriction enzyme can only occur once in the genome of the plasmid. That's what defines a cloning site. A cloning site can only occur once, because obviously if it occurred twice, I would cut out my plasmid. So you need to be able to linearize your plasmid with one restriction site. In the process of linearizing, you would end up with your restriction enzyme on either end, and now you can clone your piece of DNA. It has to be able to replicate independently of the host chromosome, so it needs an origin. And we talked about the types of origins that are found in engineered plasmids. They have such that they get multiple copies of the plasmid. They need a selectable marker, which is usually antibiotic resistance. Hence why we've knocked out any ability of these plasmids to go cell to cell, and we've also disrupted all the genes in E. coli that allow it to survive naturally in the environment, because when you clone genes, you use antibiotic resistance genes so that you can plate your E. coli on in the presence of antibiotics. If we didn't knock out those things, we would be creating more and more strains that were resistant to antibiotics. And that allows them to be very easy to isolate and manipulate. By the way, bacteriophage can also be used for cloning, and they need to fulfill these same characteristics. So you will actually not be involved in isolating the plasmid, but you can isolate plasmids from bacteria quite easily. The classic way is called an alkaline lysis procedure. As its name would suggest, it lyses the bacteria in the presence of alkaline conditions. Okay, so again, you add EDTA. EDTA is a chelator of divalent cations, and what I naturally in the cell, the outer membrane of E. coli is stabilized by divalent cations. So you can stabilize the outer membrane, and that assists in the isolation. And once you've isolated the plasmid, you can cut the DNA with restriction endonucleases to generate fragments. So who makes restriction endonucleases? Molecular cloning companies make restriction endonucleases. Who naturally makes restriction endonucleases? Well, they will make them, yes, but all bacteria, eubacteria as well, make restriction. It is their immune system. They make restriction endonucleases that recognize DNA that have patterns of methylation that are different from them and chew them up. So they don't have the immune system that we think of, but the restriction endonucleases protect the bacteria from foreign DNA invading. So basically, we have, again, made use of the natural production of restriction endonucleases that recognize specific sequences that are not correctly methylated in a palindromic form that allow us to do the classic analysis we do in a laboratory with restriction endonucleases. So it is a natural process for bacteria. So if you worked in a laboratory, how many of you have done B12? Okay, so a fair number of you. So what is one of the examples of the classic enzymes you use? It would be EcoR1, which is actually shown here. So this is an EcoR1 site. Where do you think EcoR1 originally came from? E. coli. So the name of the enzyme tells you the bacteria it came from. They're not random letters. They may seem random to you. Why is it NH1? Why is it ND3? Why is it XHO1? These are silly names. Why didn't they just call them 1, 2, 3, 4, 5? Because they isolated them from bacteria originally. Now they don't. They've cloned them and they make them in bacterial cells themselves. But they were originally isolated from their natural cells. That's why they're given the names. So using this EcoR1 site as a classic example, this is a palindromic sequence, which means it can be read the same way in terms of complementary strands. So reading 5' to 3', GAA, TPC, 5' to 3', GAA, TPC. That's a palindromic sequence. It has symmetry around the middle, and actually EcoR1 cuts between H and A, and so doing forms what's called staggered or sticky ends. So there are enzymes that cause staggered or sticky ends. There's enzymes that cause blunt ends. But from a point of view of a microbiologist, that I want you to take away is not how restriction enzymes work. To understand they are naturally made by bacteria as part of their immune system to protect them from invasion from the most common thing bacteria are invaded by, viruses. All right, so the midterm covers the end of that material. So after the break, we'll turn our attention to transposons. So come back to that 20 after, and we'll start talking about transposons.